Hello. I hope you are able to enjoy these lovely bats. These are Pteropus medius fruit bats, the reservoir hosts of Nipah virus in Bangladesh. I'll be talking about SARS-CoV-2, the origins of the virus. Where did this come from? So in general, I can say we don't know. We don't know what the origin of this virus is, and particularly we don't know what the reservoir host is, and we don't know who the index patient was. The index patient, that's case 0, the first person who experienced an infection with this new pathogen. So sometime 9 months, maybe 10 months ago, this virus was living happily in the gastrointestinal tract of a bat. And it somehow made its way into the human population. And one of the key questions that people involved in zoonotic disease, that is pathogens that are transmitted from animals to humans, in our field, one of the key questions that we ask is, was this virus supercritical in bats? Well, let's take a step back. What does actually supercritical actually even mean? The word supercritical means R0 is greater than 1. And R0 is a concept that's in the media and the newspapers and discussed frequently now. So maybe it's something that you're all familiar with. R0 is the reproductive number, the average number of secondary infections that a single, that a first infected person will transmit to others. So within your infectious period, which for this pathogen is around 7 days, within that period of time, how many people do actually you go on to transmit to? R0 is usually defined at the beginning of the outbreak when the entire population is susceptible. So was R0 of SARS-CoV-2 in bats, was it able to be greater than 1 so it could take off and spread in humans? At first, we didn't think so. At first, the hypothesis was that the virus existed in bats in a state where it couldn't bind to the receptors in human cells. And then it needed to have a recombination event in another species. So the thought was that there might have been a genetic mix-up in a bridging host species. For example, a pangolin was one of the first to be hypothesized. But this paper here suggests that actually R0 may have been supercritical in bats. So this pathogen can jump directly from bats into humans. So here, this paper came out in Nature Microbiology at the end of July, and I'll just quote a couple of things. It said, its receptor binding motif, important for specificity to human ACE2 receptors, appears to be an ancestral trait shared with bat viruses and one not acquired recently by a recombination. And the lineage giving rise to SARS-CoV-2 has been circulating unnoticed in bats for decades. So SARS-CoV-2 may still be circulating out there and many others like it with the ability to transmit within humans. We know the origins of the outbreak were in around this region, Yunnan, Myanmar, northern Vietnam, and Laos. We know this because the closest sequences to SARS-CoV-2 have been found in bats in this region. So how did the virus actually get from here into up to Hubei, Wuhan? We don't know. Did it come in a human? Was a human infected here and then got on a train and traveled to Hubei, got off the train station, went to the wet market in Wuhan and then started transmitting to other people? Or perhaps a bat was taken out of a cage and transported to Hubei through a wildlife market. Or perhaps a bat transmitted this pathogen to another species in this region, and that species, then that bridging host, was then transported to Hubei. But anyhow, we do know that there was a wet market, seafood market in Wuhan, right near the train station, that was the center of the outbreak right at the beginning. This is the Chinese horseshoe bat, so the reservoir host. We don't know which species of horseshoe bats, but we know that a number of horseshoe bats have SARS-like coronaviruses, so we expect this is the reservoir. And there was a lot of speculation about pangolins at the beginning, but now we understand that pangolins probably are just susceptible to coronaviruses in a similar way to humans. So for example, they have similar pathology as humans do for this SARS-CoV-2 virus and other coronaviruses. And this little diagram here gives you an indication of R naught. Supercritical pathogen R naught greater than 1. So sustained transmission in human outbreaks potentially is possible. Many of the pathogens that my group works on actually are not supercritical in humans. They're what we call subcritical. That means that they can't take off and spread in human populations. R naught is actually less than 1. So on average, each infected person transmits the virus to less than 1 other person. And these kinds of viruses have stuttering chains of transmission in humans. So we'll see a few infected people, the virus will stutter through a small chain. It might be 5, 10, 20 people, but it always burns out. It never takes off and spreads. And that's often because people become so sick with the viruses that they're able to be quarantined, isolated quite quickly, and that they're not then within the population and spreading the virus. No, there are thousands of coronaviruses in bat populations, thousands of unsampled viruses, and the distribution of these viruses is really quite broad, especially in parts of the world where we are not sampling wildlife. For example, in the region where this virus emerged, I suspect there may be a handful of biologists working on bats. So we probably don't know very much about their populations, about the stresses they're under, about the viral infections in those populations. There's certainly nothing that's published in this region in terms of sampling bats over and over again to understand the infections. There's a little bit of viral discovery work, but that's it. But we don't know much about the bats. We don't know about the viruses, and there's probably a great diversity of pathogens that are still out in nature. So of the bat coronaviruses, we've had 3 spill over into humans from bats in the last 17 years, starting with SARS Classic. That was the original SARS, also called SARS-CoV-1. That caused an epidemic that killed just a little over 700 people. There's MERS, Middle East Respiratory Syndrome, which is spilling over from camels into humans now. So it's gone from bats into camels, become endemic in camels, and now camels give it to people in the Middle East, and we see sporadic outbreaks there. And now SARS-CoV-2. Another 4 coronaviruses that originated in bats circulate as common cold viruses. So we are infected with them probably almost yearly. And they probably spilled over from bats a very long time ago in evolutionary terms, and then they've attenuated as the host viruses have co-evolved, and also our populations have built up immunity to these. And we're now discovering that immunity to these common cold coronaviruses may affect the epidemiology of SARS-CoV-2, in that, for example, T-cell immunity, there's been a lot in the media about T-cell immunity, as maybe attenuating the disease from SARS-CoV-2. So let's have a look at these. Let's look at these pathogens in terms of our understanding of pathogen spillover. So firstly, let's start with SARS Classic. I have an extra 0 there. That's over 700 deaths, over 8,000 cases. This was started at the end of 2002 and throughout 2003. And as far as we know, although we'll never be certain here, there may have been just a single spillover. It was then spread through civet cats throughout the wet markets in China. And it's possible there were more than one spillover, but it maybe perhaps one spillover, given the low genetic variability at the beginning of the outbreak. Ebola, West Africa. And 2014, started December 2013 in Southeast Guinea, spilled over to a 2-year-old boy who, as best we know, the best story we know is that he was playing in a hollow log, hollow tree, and bats roosted in that tree. He may have been patient 0, so the index case of Ebola, and then that spread across West Africa, as you know, throughout 2014. So Ebola, over 11,000 deaths, over 28,000 cases, a single spillover event. SARS-CoV-2. This is very, very out of date. Sorry, I did not update this. Well, when I say out of date, maybe like a month or so. Certainly those numbers actually look very different right now. And as far as we know, and given what we understand from the genetics of the virus at the beginning, a single spillover event from a bat to a human, and perhaps through an intermediate bridging host. So it gets very difficult then to really deconstruct this process of how does that pathogen go from the bat to the human when we've had 3 spillover events of coronaviruses. And given that, it's very easy to think, well, it's this simple event, right? It's just this virus, it just jumps from the animal to the human, that's it. And that's often how it's thought about, as a simple process of spillover. But what I'm going to show you is it's anything but simple. But to understand this pattern, I'm going to actually move away from SARS-CoV-2 for a moment and now transition to a couple of other viruses that come from bats, but that spill over into the human population regularly. So these pathogens, Hendra virus in Australia, Nipah virus in Bangladesh. And if you look here at the east coast of Australia, and then look at every dot here, this is a map over space and time. Every dot is a spillover event of Hendra virus from bats into horses, which are the intermediate host here. And then if we go down here and look at Nipah virus, this is Bangladesh here. And then this is the equivalent plot where we've got time here. Each little dot here is a spillover event of Nipah virus from bats into people. So here is a system where we have lots and lots of spillover events relatively to coronaviruses. And so this is the system where we need to really try to understand this process. So this is where we are working to understand spillover. And again, this beautiful Pteropodidae bat, Pteropus medius, the host of Nipah virus, is what we're studying in Bangladesh. And the Nipah viruses, they're top 10 priority pathogens. So WHO has said that they are some of the most likely pathogens to cause a pandemic, and they lack countermeasures and vaccinations. And in some places, like in Bangladesh, where this video is taken, in some places like in Bangladesh, spillover is occurring every year, multiple outbreaks, and every time we have a spillover from a bat into a human, we're rolling the dice. So far, every time it's happened, we've had those stuttering chains of transmission, that's subcritical, we call it subcritical transmission, so it hasn't taken off and spread. But can you imagine a pathogen in this environment where people are so crowded, multi-generations in one house, a poor public health system? Could you imagine if something like Nipah virus, there was a genetic variant of Nipah virus that had some pre-symptomatic transmission, like SARS-CoV-2, or maybe some people are asymptomatic. Could you imagine controlling it here? Now, one big difference between Nipah virus and the SARS-CoV-2 is that Nipah virus, the fatality rate in outbreaks so far has ranged from 50 to 100%. So 2 outbreaks in India recently had an outbreak, a fatality rate of about 90%. So 90% of people infected died. So these pathogens are scary, and we need to be understanding them. We need to be paying attention. And we study these so that we can then also understand pathogens like SARS-CoV-2 and prevent spillover from happening in the first place. So let's deconstruct spillover. So it isn't this simple passage of a pathogen jumping from animal to human. It's actually a complicated process, and there are many, many, many different factors involved. It's a hierarchical series of events, and all of these events happen at different scales, from molecules, happening within cells, they're happening even at landscapes. So I'm going to now deconstruct this a little bit here. We have things that have to happen in the reservoir host. So the reservoir host is the animal that maintains the virus or the pathogen. So in this case, bats. So we know these pathogens probably don't cause any disease in bats, and they circulate through their populations. They're endemic viruses. So firstly, we have the bats here. We have to have the bats present. For example, for SARS-CoV-2, Southeast Asia. For Nipah virus, it's actually more Southern Asia. We have to have them at a density that is high enough to be able to maintain the pathogen. That pathogen has to be released from the reservoir host. So for SARS-CoV-2, we know it's released through feces. So bats actually shed the virus in their feces. For Nipah virus and Hendra virus, the bats shed the virus in their urine. For Ebola virus, we actually don't really know how it's shed from bats. The pathogen then has to survive and be able to survive in the environment for long enough for the next host to become exposed. So here I have a human exposure, but it could be for a horse for Hendra virus, or it could be a civet cat for SARS-CoV-2. But that next host actually has to be exposed, and then that next host has to be susceptible to the infection. So that means the virus has to be able to bind to the cell. It has to be able to get into the cell. It's got to be able to replicate, exit the cell. It's got to be able to evade the host innate immune system and then disseminate throughout the body of that host. So we can think about it as an infect, shed, spread paradigm. So we have to have an infected animal, it has to shed the virus, and this is all happening in the wild animal host, and then it's got to be able to spread it to humans, and then we also have the human-to-human spread component right here. And what makes this really challenging to study is that all of these different components are, many of them actually, not all, almost all of them, are very dynamic in space and time, so constantly changing. We have bats that are nomadic or migratory, moving across the landscape with different densities at different times of year. The pathogen within the bat populations is also very variable in space and time. It will pop up here and then disappear across the landscape. So very hard for us to study pathogens that are so dynamic in both space and time. And then we also have like the dynamics of, for example, susceptibility in the human population and so on. So very, very changing, constantly changing. And all of that, actually, all of these conditions have to be able to line up at the one point in space and time for the pathogen to be able to overcome all those barriers to make that species jump there. I'll give you one example from a personal experience of spillover in Australia. Again, back to Hendra virus. So this is a Nipah virus. And I think I failed to say before, Nipah viruses are in a genus within Paramyxoviridae. Paramyxoviridae, the paramyxoviruses, you will all know the paramyxoviruses. Measles is a paramyxovirus, mumps is a paramyxovirus, canine distemper is a paramyxovirus, rinderpest. So one of the 2 viruses to ever be actually eradicated. Rinderpest is a livestock disease. It's a paramyxovirus. So Hendra virus in Australia is endemic in the very large bat species. They look very similar to the Pteropodidae, Pteropus medius that I showed you in Bangladesh. They live very happily with this virus, no disease, no signs of pathology. And when a bat is feeding in a tree in a horse paddock and then excretes virus in the ground, probably through urine, and a horse comes along and grazes on that same patch, the horse then can become infected. And in 2017, there was a cluster of these events in the east coast of Australia. My team actually visited one of the properties that was infected. The owner of the horse was traumatized. We came a few months after the event, but as she retold the event, she had tears. I suspect she had post-traumatic stress disorder from this awful, awful thing that had happened to her. So this is what happened. It was raining. Curiously, veterinarians who deal with Hendra virus often say it was raining during the outbreak. So it was raining. And because it was raining, her horse's paddock turned to mud. So if her horse's paddock had not turned to mud, she would not have moved her horse from the paddock into an orchard. Within the last 6 months, a new population of flying foxes, so there's Pteropodidae, big fruit bats, had moved into a neighboring property. And this is something we've seen in Australia, new populations, small populations of flying foxes popping up in urban areas and agricultural areas because they've lost their critical winter habitat. So these new bats, they popped up in this area, and those bats were having a nutritional stress event, so a starvation event at the time. And because of that, they came into the orchard where the horse was now kept to feed on lemons. Lemons are a food of last resort, a starvation avoidance food for bats. Because the bats were starving, they were shedding Hendra virus. And our team was we were catching bats and measuring Hendra virus in that region at the time. We found very, very high levels of Hendra virus. But now we understand that there's an association between nutritional stress causing the bats to be immunocompromised. And because they can't control the pathogen, they're releasing the pathogen in their urine at much higher levels. So the bat was shedding Hendra virus. Because it was raining and cold, that virus shed onto the ground was probably able to survive really well for a period of time, certainly for long enough for the horse to then come along and be exposed to the virus, to an infectious dose of Hendra virus. And the result of that was the horse was killed by this virus. It's very acute onset of infection, 2 days, a frothy nasal discharge, neurological symptoms. Horses are usually euthanized because it's such a terrible death. This was her family pet. This was a horse that was trained to ride without a halter. It was really a part of her family, and so an incredibly traumatic event. Her kids were exposed. They had to be tested every second day to this pathogen that has a 57% fatality rate in humans. Her kids were okay, she was okay, but it was a horrible situation to happen. But as we stood in the paddock, we're actually standing right amongst these lemon trees as she told us the story. I could just see all these layers go ching, ching, ching. And I was there with co-authors of this paper that we wrote. This was in Nature Reviews Microbiology, and it had just come out actually. And we looked at each other and we're like, wow, yeah. So the reservoir host distribution had changed to align with horses. The bats had come into the agricultural area. They were they were infected with a high level, high prevalence. It means proportion of animals in the population infected was high because of this nutritional stress event. They're releasing the pathogen, shedding it in their urine because of probably their immune status was compromised. And then, of course, this cold weather allowing the pathogen to survive in the environment. And then this horse behavior of sniffing around under these trees and becoming infected. The different pathogens have different spillover pathways. Hendra is similar to Nipah genetically, but Nipah has a different epidemiology. So Nipah virus in Asia is transmitted either directly to humans or through other species. In 1997, there was an outbreak of Nipah virus in Malaysia, where bats came in to feed on fruit trees that had been planted next to pig farms. The fruit that when bats feed on large pieces of fruit, they like to chew up the fibrous material. They take out all the juice and then they spit the rest on the ground. And what we suspect is the pigs then picked up those spats, we call them spats, on the ground, and then became infected with the virus. And then Nipah virus spread then among pig populations throughout Malaysia as humans moved them around. And 200 people were killed by Nipah virus. But in Bangladesh, we see the outbreaks every year, and I'll talk about that in a second. Then in India, which was actually where this photo was taken, there have been outbreaks over the last couple of years, but we don't know what the spillover mechanism was. In Bangladesh, it happens because of date palm sap collection. So here is a date palm sap collector. He's climbed up the tree. He makes a big gash in the tree, as you can see here. And then the date palm drips down, and through this straw drips into the pot. And I haven't actually tried it, but I was so tempted to try it when I was in Bangladesh. I thought that's probably not a good idea, but it sounds like it's really delicious. In the morning, this is sold on the street, but this is what happens during the nighttime when it's being collected. Here's again, Tropus medius. This bat is licking the date palm sap as it's running down the tree. And I want you just to have another look here. I'm going to go back to the start. Notice the bat, while it's feeding on the date palm sap, turns upside down and pees right into the pot. And then that pot is sold fresh the next day. And date palm sap's a really nice medium for allowing a virus to survive. Now, outbreaks of Nipah virus have been actually traced along the roads and of the date palm sap collectors. So there are maps showing that this date palm sap collector sold pots all along this road, and there were Nipah virus outbreaks actually all along that road. So it's actually a really effective system for getting a virus from a reservoir host into a human with devastating consequences. So how do we study those top parts of the spillover process? So the bats, the virus in the bats. Well, this here is a graduate student in my lab. This is Devon Jones, and she led our field teams in Australia for a time. And I'm going to show you a video here of she is and the team catching a bat in a net. The bat has just come back, it's been out feeding all night, and as it's flying back into its roost site, it hits the net, and we pull the net down, untangle the bat. And this bat now doesn't want to let go of its crocus palm fruit. It's actually a weedy species and another one in an agricultural area. Then we anesthetize each bat with a gaseous anesthetic. We take this tiny residual molar from each bat to check the age. So we count the rings in the molar just like tree rings. And that gives us a lot of information for disease work. We take a blood sample. That blood then comes back to MSU, where we do a whole suite of immunological assays to understand the immune status of the animal. We look at blood-borne bacteria on these FTA cards you can see here. We take urine. Urine is really important to us because that's where we see the viruses. The Hendra virus is shed in the urine, but we also do a full urinalysis. There's a whole bunch of other things that we do, information we gain. Then we release the bats back into their colony. And this is a roost site actually in an urban area, one of those new roosts that I talked about a second ago that have just popped up recently. So this is a picture of all this information we take from every individual animal. So we go back to the same populations again and again through time to build up data sets and patterns of how the virus is circulating, what is the immune status of this animal over time. And this is what the data looks like. So here we have a prevalence, that's the proportion of the population infected over time. And you can see that prevalence changes over time. These shaded regions are winter. So we see higher prevalence in winter. This dot here is actually that population right near that horse I told you about. So that population had around 60% prevalence at that point in time of that spillover event. So you can see that that's a really important part of spillover, is how much virus the bats are contributing, how much they're shedding into the environment. So this is actually aggregated over a whole bunch of field sites. But as I said, we have many sites and we go back month after month after month, and we see what patterns emerge over time. When do we see more virus? When do we see less? And this allows us then to say, what are the factors driving high prevalence in populations at a certain time? So one thing that our group has found, and this is really relevant to SARS-CoV-2, this was led by Alison Peel at Griffith University. This is time, and these are the number of viruses. Each color is a different virus found over time. This was actually in a huge cluster of Hendra virus outbreaks in 2011. They found a lot of Hendra. So again, it looked actually something like this in 2017. It looked like that, lots of Hendra virus. But bats shedding Hendra virus was shedding lots and lots of different pathogens, all these different paramyxoviruses here. And what that tells us is that it's likely that those bats were all experiencing something affecting their immune system that was then allowing them to shed all these pathogens that they happen to be carrying or happen to be circulating in their population. And that's what we actually think is happening with SARS-CoV, with the coronaviruses in bats. The very little bit of evidence, there's 3 longitudinal studies, 3 studies like this that would look like one population, but nothing that looks like many populations. But 3 studies of one population of coronaviruses in different bats shows that there is more or less shedding throughout the year, and that there's more shedding when the bats are at poor body weight, so perhaps in poor body conditions. So maybe this nutritional stress hypothesis is relevant. And also when they're co-infected. For example, bats here with white nose syndrome, there was an experiment showing them shedding 60-fold more coronavirus when they were infected with white nose syndrome. That was experimental data. So as well, we also want to know about the behavior. So remember, in that diagram, there is the overlap of the reservoir host and that recipient host. It's really important. So this is work actually by both a PhD student in the lab, Maureen Kessler, and a PhD student at Texas Tech, Adrienne Dale, looking at the behavior of bats. And what they've found is during food shortage events, the bats are more likely to feed in horse paddocks. So that behavior is critical. Is there something maybe in SARS-CoV-2 with horseshoe bats where they may be seeking resources more in human environments at certain times of the year? That would be something that should be studied. So I think this is now a reasonable overview of all these components of pathogen spillover, starting with the reservoir host. So for SARS-CoV-2, we know the reservoir host is the horseshoe bats, but we don't know which species. We know that they're in that area of Southeast Asia around Yunnan province, southern China, northern Vietnam, Myanmar, Laos. We know that prevalence changes in space and time, so we'll see highs and lows of prevalence, but we don't know anything about the dynamics of those viruses in the bat populations. We know those coronaviruses are released in feces, so that's why people have thought maybe guano mining could be a part of this, but it's also possible that bats came into wildlife markets and feces was a way of spreading the virus. We know SARS-CoV-2 survives very well in the environment, at least for hours under reasonable conditions. And we know that it's possible that in its form in the bat, it was supercritical, meaning it actually could get through all of these layers. We know, for example, that the virus can replicate within mucous membranes. So if you touch your eyes or mouth or nose, it can bind to cells in those areas. We know that it has an affinity for the ACE2 receptor, so it can actually get into human cells. It can replicate in those cells. It can assemble itself and bud from those cells. We know it can evade the innate immune response and disseminate within the human body to then be spread through the human population. So that's really a lot of big questions in the spillover of SARS-CoV-2, and that's really the state of our knowledge right now. Thank you very much.